Hello, good morning, and thank you all for coming. And it's good to see you once again. So today I present a scientific presentation, and it will be on UV inactivation, which is delivered by my company and was developed by my company. So it is based on two scientists, two medical doctors, myself and Daniel, who is sitting with us today, and we were classmates at the medical school. And in the end, the COVID-19 hit, and we created new opportunities to enable science, enable BSL-3 and BSL-4 science outside of those labs to create more capacity to pandemic response, pandemic preparedness and rapid response technologies. We are now teaming up with large companies and large entities having wonderful projects, and we are now growing. We have a beautiful team now of an experienced project manager and lead advisor, Tibor János, who is a legal advisor and a very experienced IP manager, and also we've got Peter, who will be our sales director, which is exciting. So you know all the kinds of biosafety levels, of course, but this is really the key problem here, because as you know, there are a very limited number of BSL-4 labs in the world, a bit more BSL-3 labs in the world, but the demand to run projects in these kind of labs is ever increasing. And these are the main components of pandemic preparedness, these labs. There's only 51 BSL-4 labs throughout the world, and of course we're quite happy to have one here in Pécs, which is a regional racer. So our technology basically is the UV inactivation of viruses. You can imagine them by imagining a remote control for the tally. You just switch them off. And all the advantages come from this switching off of the virus. So the viruses are, of course, infectious. They create a great problem, and by being infectious, you have to work with them in a high containment lab, very restricted area. It is highly expensive, and this is what we want to avoid by creating a UV inactivated version, which is apparently the same without the infectious risk. And you can use them as model services or substituaries for the active virus itself. So this is, in one slide, the advantage that we can skip these levels while maintaining viral integrity. And this is a project that we won a large grant for to take a leap, take a great leap in this kind of technology, to go to the industrial level. And I want to show you some results, because it sounds quite all right, yeah? Inactivation, it sounds an enabling technology. But the really great leap was when the Hungarian Defense Forces investigated the virus that we had. And what you can see with red is the active virus. It's an ELISA assay to the S protein. The blue charts are the UV inactivated bars. And as you can see, even the statistics say that there is no change. So you can't detect any changes on a protein level, which is amazing. We wanted to do diagnostic tests. We wanted to develop new technologies and vaccine development based on UV inactivation, and this is the core slide that enables us to do that in a safe way with the protein and the viral integrity. And another one, which really surprised us, was done by the National Lab of Virology, and it's the PCR. So we performed PCRs, and as you can see, we compared, the red is again the active virus, and the blues are increasing dosage with UV, and we didn't detect any change around the minimal effective dosage in PCR. And we performed that with Zika. We inactivated Zika virus. We inactivated the West Nile virus. We inactivated another few. And for PCR, we experienced that there is no change around this area where the virus is already inactivated, but you can't detect any change with PCR or with ELISA assays. And of course, we had to move a lot through biosafety. This is no easy technology. We had to make sure that these virus assays are indeed inactivated and completely safe to use outside a BSL-3 or 4 lab. So we had to make sure. And we wanted to make in vivo tests, which we did. And on the third slide, you can see the body weight change with gray for the control and green for the UV inactivated one and the red for the active one. And we saw that throughout a week with the hamsters, there was a body change only with the active virus. Okay, it seems okay. There is no systemic change when the UV inactivated virus is introduced into the system. And we also did a nasopharyngeal PCR. It was a ddPCR, and we found that, okay, there is no replication inside the animal. And we also developed an ELISA assay to measure whether this UV inactivated virus could provoke any cellular immune response. And what we found was amazing. We found that it was similar. It was an intranasal spray to the hamster. What we found is that it was similar to the active virus's immune response. So what we created here in COVID-19, this measurement was done in 2021. So we created intranasally a first-generation vaccine-based material. It was amazing. So a few remarks. So using UV inactivated agents and viruses will allow us to minimize the risk. So I've got this point really clear that we only need to use high containment labs as amazing tools, but when only needed. When we only want to do the active virus research in those high containment labs. And I want to introduce a proposed 3R principle for high containment labs. So if it's possible, we need to use the animal experiments 3R for HCLs, whereas we need to replace the viruses, where it's possible, with UV inactivated forms, because it will allow the researcher to do almost the exact same experiment, if it's a non-propagated work, with the UV inactivated one. And thereby you can reduce the number of necessary high containment lab entries and minimize the associated risk and optimize the efficiency. So just imagine that with one lab entry into the BSL-3 or BSL-4, you could generate loads of UV inactivated viruses and do the research outside proficiently and safely and cheaply, to be honest. And it will allow you to minimize the risk and do it in a safe environment. And we want to take the leap here. We are taking the leap to the industrial level. We are creating a technological pipeline within this project to create large volumes of inactivated viruses within one GMP system. And it will enable a very fast and rapid act in times of a pandemic, and in peace times, it will enable a safe and more affordable research. And really, it will enable researchers, for example, on the military and on the civil side, to do research with the inactivated agents. So this is really are the applications and benefits here, that we can do a CBRN research, maybe outside of these labs, and do pharma. We even had Zika experiments using the inactivated Zika virus that would allow for antiviral testing, because the UV inactivated viruses behave quite the same when introduced to a cell, even to the point when replication will occur. The replication, of course, stops, but until the point the virus is introduced to a cell, it is amazingly the same. So you could use that as an antiviral endocytosis testing platform. And of course, we have the demands from the WHO. As yesterday, we know what to inactivate, what viruses we have to concern in these kind of technology. And there are a few competitors on the market. This is a nice market opportunity too, because 1 milliliter of this UV inactivated virus would cost $500 to almost $6,000 a milliliter, 1 milliliter. And we have got the comparative advantage in this new taking the leap industrial section, because the CAPEX and the OPEX are lowered amazingly compared to the original and benchmark studies and platforms that you would find in the market. And these are our advantages. And we will do sales, of course. We want to grow as a company and to enable researchers to do their work. And this will be done by direct research agreements, or in the future, we want to sell the technology through a license or develop new vaccines, for example. BSL-1 is also amazing, and UV inactivated vaccines are on the rise. If you see the articles, you can see more and more UV inactivated vaccine models and platforms. For example, there's a human one for H1N1. This is a vector-based but UV inactivated human vaccine. And on the right-hand side, you can see a pig vaccine with UV inactivated chlamydia pathogen. So this is really it. This is our technology, and I think this technology is on the rise. Many papers are being published year by year, and we want to be the first ones to take this leap into the industry. which I thank you for the attention.